Among the most promising areas in longevity and cancer prevention research are the nutrient-sensing pathways, especially AMP-activated protein kinase, AMPK, and sirtuin-1, SIRT1, which serve as molecular sentinels that detect shifts in energy availability and cellular stress. These pathways do far more than simply react to food intake. They orchestrate broad protective responses that modulate metabolism, reduce inflammation, activate autophagy, and support genomic stability. As we age, disruptions in AMPK and SIRT1 signaling contribute to the breakdown of these defenses, leaving cells more vulnerable to degenerative diseases, including cancer. Yet, by understanding and potentially reactivating these pathways, science may offer tools to slow aging, enhance autophagy, and suppress tumor development. AMPK is the cell's primary energy sensor. It becomes activated in response to low ADP levels, such as during exercise, caloric restriction, or fasting, prompting the cell to switch from energy-consuming processes to energy-conserving and repair-enhancing ones. One of AMPK's key actions is the activation of autophagy. It does this by directly inhibiting mTOR, a nutrient-sensing kinase that suppresses autophagy, and by activating ULK1, which initiates autophagosome formation. In aging tissues, where nutrient signaling becomes dysregulated and oxidative stress builds up, AMPK activation helps restore metabolic homeostasis, clear damaged components, and prevent cellular senescence. In doing so, AMPK reduces not only metabolic dysfunction but also the buildup of DNA mutations and reactive oxygen species that promote tumorigenesis. SIRT1, another central longevity regulator, responds to changes in the NAD/NADH ratio, an indicator of cellular redox and metabolic state. Like AMPK, SIRT1 is activated during periods of low nutrient availability. It enhances autophagy by deacetylating key autophagy proteins such as ATG5, ATG7, and LC3, facilitating their activity. It also exerts anti-aging effects by maintaining mitochondrial health, supporting DNA repair, and suppressing pro-inflammatory gene expression. In the context of cancer, SIRT1 has shown both tumor-suppressive and context-dependent tumor-promoting roles. In normal cells, SIRT1 activity maintains genome integrity and prevents malignant transformation. But in some cancers, particularly those already established, SIRT1 may help tumor cells survive metabolic stress. This duality underscores the need for precision when modulating these pathways therapeutically. Importantly, AMPK and SIRT1 do not operate in isolation. They form a synergistic network. AMPK can increase NAD levels, thereby promoting SIRT1 activity. Together, they work to suppress mTOR signaling, activate autophagy, and promote mitochondrial biogenesis through PGC-1A. This axis, AMPK, SIRT1, PGC-1A, is often referred to as a master regulator of energy metabolism and healthy aging. When fully functional, it supports a resilient cellular environment that resists both age-related decline and the metabolic reprogramming that cancer cells rely on. But with age, these pathways lose efficiency. NAD levels decline, reducing SIRT1 activity. Chronic nutrient excess, inflammation, and mitochondrial dysfunction inhibit AMPK signaling. Autophagy becomes impaired, oxidative stress rises, and cells enter senescence or accumulate mutations. These changes are not merely correlated with cancer, they are causative. Senescent cells, with their pro-inflammatory secretions SESB, create a tumor-permissive environment. Impaired autophagy allows damaged DNA and organelles to accumulate. Dysfunctional mitochondria leak ROS, increasing genomic instability. The erosion of AMPK and SIRT1 activity is thus a slow unraveling of cellular defenses against cancer. This makes AMPK and SIRT1 powerful targets for therapeutic interventions. Activating these pathways pharmacologically or through lifestyle changes may restore youthful cellular function and reduce cancer risk. Caloric restriction, intermittent fasting, and exercise all stimulate AMPK and SIRT1, boosting autophagy and mitochondrial health. Pharmacologic activators like metformin, an AMPK activator, and resveratrol or NAD, precursors which enhance SIRT1, are being explored not just for metabolic disease, but for their anti-aging and anti-cancer potential. In animal models, metformin reduces cancer incidence and extends lifespan. Resveratrol and NAD Boosters have shown similar effects in delaying age-related degeneration and improving autophagy. Still, these interventions are not without risks. In individuals with undiagnosed cancers, activating autophagy or boosting cellular stress responses might inadvertently help cancer cells survive. This reflects the persistent challenge of duality in biological pathways. What supports resilience in one context may support persistence in another. Moreover, prolonged activation of autophagy without adequate control mechanisms could lead to excessive degradation or even autophagic cell death. The key is balance and timing, especially in older adults whose cellular landscapes are often a patchwork of healthy, senescent, and potentially malignant cells. In summary, AMPK and SIRT1 represent critical control points in the intersection of nutrient sensing, autophagy, aging, and cancer. When functioning optimally, they reinforce a protective network that resists metabolic imbalance, clears damage, and preserves cellular youth. Their decline with age is not merely a symptom, but a cause of dysfunction and disease. While there is significant therapeutic promise in targeting these pathways, especially through lifestyle or pharmacologic means, precision is essential. Future medicine will likely depend not on whether we can activate AMPK or SIRT1, but on how, when, and in whom, tailoring interventions to each individual's age, metabolic state, and cancer risk. In doing so, we may find not just a treatment, but a path toward healthier aging itself.